deliver the welcome address. Over to you, sir.
Thank you so much. Good morning to all. The blessings of Almighty. I also would like to salute our patrons, Chancellor Dr. J. Ramachandran sir and Pro-Chancellor Dr. Rajesh Ramachandran sir for their generosity. I cordially welcome our Vice Chancellor, Colonel Dr. G. Srivastava sir, the man of inspiration. And my humble welcome to our beloved Registrar Dr. N. Jayaprakash sir for his untiring support and encouragement and the man began the show. It's my great honor to welcome the guest speaker of this webinar, Dr. Murali Melligaraman, Regulatory Toxicologist, Chennai. Welcome, sir. I also extend my hearty welcome to the most respected Joint Registrar, Controller of Examination, Deans, heads of the various departments, directors. A warm welcome to you all. A cheerful welcome to the participants, my colleagues, and my dear friends. It's my great honor to welcome the dignitaries on the screen and off the screen. The workshop provides a platform for researchers, students, and faculties to understand elaborately on the molecular techniques which involve in assessment of aquaculture health. The workshop includes two eminent speakers. They will share their experience with two unique topics, which ultimately brings us the enormous information which is going to be improve participant knowledge of the molecular aspect. Okay. Thank you all for giving me this opportunity. Let's land it there. Thank you.
Thank you, sir. May I now invite Dr. Maya Kannan, sir, Organizing Secretary, to introduce our today's speaker. Over to you, sir.
Thank you, Ms. Swarna. Dear speaker, participant, and my colleagues, a very pleasant morning. It's great privilege to have this opportunity to introduce our guest speaker of this session, Dr. Murali Malligaraman, Regulatory Toxicologist, GLR Laboratories Private Limited, Chennai. Dr. Murali has attained his doctoral degree and PG degree in biochemistry from University of Madras, India. After his doctoral degree, he entered the pharmaceutical industry and involved in the development of drugs from natural sources. Then he moved to University of Malaya, Malaysia, as postdoctoral researcher, where he shifted his focus into regenerative medicine for the treatment of orthopedic disease. His commitment towards research and teaching earned him the academic position as an associate professor in the same university. Dr. Murali worked as several projects with international collaborators. He is a prestigious author of many articles, book chapters, and papers in peer-reviewed international journals. He is the editorial board member and reviewer of many academic journals, and he has given a number of lectures both in India and abroad, and talks in national media, including COVID. On a final note, Dr. Murali Malligaraman is a fascinating individual, always a researcher and mentor for students, scholars, not only his own, but also from other institutions. Sir, it's a great honor to have you here for this lecture. Over to you, sir. Thank you.
Thank you, Dr. Maikanan. Thank you for the wonderful introduction. Just give me a minute. I'm just sharing my screen.
Yes, sir. Please share. Did you share already, sir?
Um, Something buffering, I believe.
Sir, you have to share your entire screen, sir. Then only the PPT will be presentable.
Okay. Just give me a minute. Something is not responding here.
No problem, sir. You take your time. Sir, if you have a problem to share it, you maybe share the PPT. Yeah, it's visible, sir. Thank you. It's visible. Sir, your mic is muted. Murali, sir, your mic is muted, sir.
Now it's fine, right?
Yeah, you are audible now. Thank you, sir.
Okay. Then just okay. Thank you, one and all. I really thank the interest of each and every participant and the organizers to even working on the Sundays. Because usually the Sunday is the only one day we say like we used to get rest. But unfortunately, due to the lockdown, we have a long rest and we are trying to cope up with the time in a very useful manner. And I'm really thankful to the organizers of this workshop and to the Ahmed University for making their students and staff to be active during the pandemic. So without any further things, I will just directly enter into my topic of presentation today. Today for next maybe like 40 to 50 minutes, I will be talking about few basics of PCR, polymerase chain reaction, and also we'll be touching about the people who has introduced me about the PCR and where I applied the PCR and where the PCR can be applied towards the aquatic health and aquatic toxicology. It was nearly 38 years, almost we have going to cross 4 decades after the discovery of the polymerase chain reaction by Dr. Carey Mullis. Science is fun, science is interesting, and science is multidisciplinary. You see, Carey Mullis, he did his basic degree in chemistry, but however, he got a PhD degree in biochemistry. I'm happy that I'm also a PhD degree in biochemistry. And with the basics in chemistry, completing a PhD in biochemistry, he has introduced a groundbreaking technology. He has developed a novel technique, the PCR, which has made a revolutionary things in the field of molecular biology, genetic engineering, and biotechnology. So that's why before starting my the talk itself, I want to emphasize one thing. Science is not standalone. Science is multidisciplinary. Science is interrelated. That's why we people are interacting even nowadays through online. We are interacting between different industries, different academics, academic and industry collaborations. So science live only by discussions, only by debate, and then only we can find out many new things. So we already have passed nearly 40 years, but when Carey Mullis was introduced the PCR, uh, and this over these 40 years, around this 38 years, maybe in the 36 years, the PCR technique, the polymerase chain reaction techniques, was only known to the scientific community. So we used to use in different words, so we confuse the common layman, right? We used to say chromosome, gene expression, transcription, translation, nucleotide, adenine, thymine, guanine, cytosine. How much of complicated things we used to complicate the common man. So only the scientist or the student who has get used to the PCR know what a PCR is, the polymerase chain reaction is. But because of the pandemic, even today, even a common man knows what a PCR is about. Even a common man, common people, whomsoever even walk into the road, stop a person, ask, oh, what is PCR? You'll say, oh, I know PCR. PCR is a diagnostic tool for the COVID. So think about the evolution of the PCR. Started from a simple technique, when only the scientists know about it, now each and every layman of the country of the world knows what PCR means. Of course, they know PCR only as a diagnostic tool. But we scientists and we researchers, we academics, we know the application of scientists in various aspects. And what I'm going to do today is just touching a few aspects of that. Due to time constraint, we cannot be able to go and discover each and every portion of it. But I hope I will cover the major portion of it, and even if something is left out, we can take it further in the later sessions. So as I said, like each and every common man knows what a PCR is about. Day to day, we are hearing the news. There is a number of cases this, number of cases this, 30,000 cases. Today there is a decrease in the case, 29,000. So all these number of cases we are hearing in the news is the outcome of a PCR result. So everyone who is either affected or who is suspected to have a COVID-19 is undergoing a PCR test, and whatever the numbers we are seeing in the news media is only the outcome of the PCR result. So as I said, just 1 year back, maybe in 2019 December, PCR is known only to academics, researchers, scientists, and students. Now, even a common layman, even I think my wife, my father, even anyone can, your father, your son, anyone can talk about the PCR, as there are much amount of things highlighted in the news media about the PCR. Now, every layman knows PCR is a diagnostic tool that's used for the detection of COVID-19, and even one step ahead, they know what CT values. Even though they doesn't know the technicality of the values, what CT means, they know that if the CT value is higher, means non-infectious. If the CT value is mild infection. If the CT value is low, then it's highly infection. Of course, they don't know how to interpret the CT values, but we know, right? When the CT value is higher, it means that is low viral load. That's why we are seeing the peak after the 40th cycle or something like that. If the CT value is very low, maybe 50 and 20, which means that the SARS-CoV-2 viral load is higher in the subject. That's why we are getting the lowest number. So researchers know what's the technicality between the lower number and higher number. But even a common man knows that if the PCR results say that the CT is 40, he says that, okay, I'm happy, I'm negative for COVID-19. If the PCR result says that 50 to 20 CT value, he says, oh, I am in high risk. So at least that kind of knowledge a common man already has over the PCR test. Okay, we always say Mata, Pita, Guru, Deva. So considering my guru, remembering my guru on this day, Professor Dr. S. Niranjali Devaraj. Of course, every layman you meet in your life is going to be your teacher. Either they are going to teach a good thing to you or a bad thing to you, but definitely the person is going to be a teacher. So we came across many teachers right from our elementary schools, higher secondary schools, colleges. But on this day, why I need to specially remember my guru, Dr. Niranjali Devaraj, is probably because she is the first person who might have introduced me about the PCR. That may be probably in the year of 1998, while I was doing my master's degree, master's in biochemistry. Maybe in the master's first year, maybe in the molecular biology course, she is the first one who is going to be introducing, who has introduced me about PCR. But those days, it's popularly known as thermal cycler. Then the term evolved as PCR, that's polymerase chain reaction. Then we had multiple terms, qPCR, quantitative PCR, real-time PCR, multiplex PCR. Of course, right from thermal cycler, PCR, RT-PCR, and multiplex PCR, I have went all this journey, starting from a simple semi-quantitative PCR to up to a place where even I can able to do the multiplex PCR. But however, all this is achievable only by a single person, a professor, who has taught me about the PCR techniques. So whoever is listening here, please be thankful to your teachers. They are transferring the knowledge to you which they already have. So without our gurus, our teachers, we are nothing. So what is PCR? So PCR is known as a polymerase chain reaction. The polymerase chain reaction is a biochemical technique that uses the thermocycling and enzymes to quickly and reliably copy a DNA. So what we are doing is just a biochemical process which is going to copy a DNA. Why this is required and why the thermocycling is important. So as I said, in a PCR, the earlier terms is only thermocycling. Means, I hope like everybody has at least seen the PCR machine in the laboratory, because nowadays the PCR machines are most common in each and every laboratories. Those days, while we are doing our masters, I remember we have never seen the PCR machine. While we are doing our PhD, the PCR machine is available only at the certain labs. If the PCR machine is available in a lab, we consider that as highly funded laboratory. So that's how it went about. Even in those days, even the real-time PCR was not common. We can able to get only the semi-quantitative PCR. Irrespective of whether it's a conventional semi-quantitative PCR, a real-time PCR, or the multiplexing PCR, the common thing that's running around in your PCR machine is only thermal cycling. So thermal is temperature, cycling is you are cycling over and over. So what is the thermal cycling that's happening in the PCR? If you see the steps in the PCR, there will be an initial step which is called as, okay, to achieve the thermal cycling, when the PCR was first introduced, Okay, remember when the PCR first introduced, now it's a single machine where you load all the sample, put it inside, close the door. After a certain time, open the door, you will get the DNA. But when PCR was introduced first time, the entire PCR reaction was carried out in a water bath because PCR requires the temperature to be cycled. 3 water bath will be made in a 3 different temperature, 90, 72, and 54, something like that. So after finishing each reaction, we have to change it manually, one water bath to other, to keep the thermal cycling. So this was happened till I think 1996, somewhere I forgot the year. But since the invention of Mr. Cycle, that's the first name given to the thermocycling machine, whatever you call as PCR machine nowadays. The Mr. Cycle is the first thermocycling machine which automatically can taking care of shifting between its temperature. So thermal cycling is the important process in your polymerase chain reaction. So why is thermal cycling is important? So if you see the process of the PCR, the steps of the PCR, your first step will be initialization, or it's called as a initial denaturation, where the DNA template will be heated to a higher temperature to denature your circular DNA, cDNA, or the genomic DNA. First step is to unwind these two strands. As we all know, DNA is made up of double helix. So first thing is we need to give a high temperature to unwind these two strands, denature the two strands. And also this step is very critical to quick start for your DNA polymerase. In most of the reactions, the DNA polymerase which is used in the PCR is stack polymerase that's acquired from Thermus Aquatic, and which can do its action at higher temperature. So the initial denaturation will unwind your DNA and prepare your sample for the quick start for the DNA process. So after the initial denaturation process, where comes your thermal cycling? Usually there will be a step of denaturation, annealing, and extension. So what is the denaturation? Your DNA has to be unwind. The annealing step is where your primers in the PCR you will be adding a specific primers. I will let you know what are the primers later. You will be adding the primers. Once the primers will go and anneal to the denature or unwind the DNA in the annealing process, annealing step. That's at the different temperature. Mostly your denaturation temperature will be 94 to 98 degrees Celsius, and your annealing will be at 80 to 64 degrees Celsius. And after the primers anneals to that, there will be a step of extension, where the DNA polymerase is going to extend the DNA, which is going to happen at 72 to 80 degrees centigrade. So where comes the cycling is, as I said, after the initial denaturation, your cDNA or the genomic DNA, whichever are the sample you are going to amplify to the PCR, is go to step denaturation, annealing, and extension. These steps will go over and over and over and over to nearly 30 to 40 cycles. So this is where the thermal cycling is happening. The temperature is cycling between 98, 50, 72, 98, 50, 72. Repeatedly it will go on again after the initial denaturation. So all that happening in your PCR machine, the PCR machine is just your water bath, which can able to quickly change between the temperatures. So only this quick change between the temperature is required for that machine to perform the PCR reaction. As I said, in the denaturation, the DNA is going to unwind. In the annealing, whichever primers you are going to add, it's going to go and anneal to the unwinded strands. After annealing, your Taq polymerase is going to bind, and it is going to take care of the DNA synthesis, where you'll be getting the copy of the DNA. Say, for example, as I said, we are going to 40 cycles. In these 40 cycles, the sample is amplified exponentially, not geometrical, it's exponential. What it means is, say for example, we are starting with one strand of DNA in the beginning. After a cycle, it will be 4. After the next cycle, it will be 4. After the next cycle, it will be 8. After the next cycle, it will be 16. So consider that your starting sample containing 1,000 copies of your DNA. After your first cycle, it will be 2,000 copies. In the second cycle, 4,000 copies. Fifth cycle, 8,000 copies. And it goes on like that. So a sample you started with very minimal is going to be get amplified to the larger quantity of detection through the polymerase chain reaction. And if you see the phases of PCR, usually there will be 3 different phases of PCR. Either it's conventional or it's real time. All these 3 phases will happen. So exponential phase, this is where the starting phase of your DNA to get rapidly copying or rapidly multiplying in your PCR machine. But however, as is the initial stage, even if it is real-time PCR, mostly it won't detect the flows and signals that's coming out because that's starting stage. So after it crosses the exponent state, there will be a linear state where you will be having a large number of accumulated copies of your DNA, Either it can be visualized in the real time as such, if you are using a real time PCR, or you can take the samples out from this and you can run an agar gel to be detected in as a band in the agar gel electrophoresis. So what will happen after the linear phase? It will reach the plateau. Why it need to reach a plateau? Because in the PCR reaction, you will be giving all the mixtures, the enzymes, the cofactors, all these things to start the reaction. So in the exponential phase, the reaction is starting so that you cannot able to see the DNA getting amplified. The linear phase, there is accumulation of the copied products so that you can able to see the response. After that time, the enzymes may be one off, the enzyme activity may be lost, or all enzymes will be utilized, all nucleotides will be added, will be utilized. So after some certain cycles, maybe if you go for 50 cycles, you won't get any response beyond a certain point. So that's why it's called as plateau. So each and every PCR reaction will go 3 different phases: exponential, linear, and a plateau. And mostly the starting point when you are getting to see the fluorescence, that point is detected as CT. That's why I say, like, if your CT, if your person CT in a COVID test is going to be very less, means he has high viral load. Instead of that, if the CT is going to start here, maybe this is a cycle, right? So if the person is going to see detected in the earlier cycles, means he has high infectivity. If the CT value is going to be somewhere around 30th or 40th cycle, mostly he will be the negative. He won't be getting anything over the source code. And detection methods, of course, I won't squeeze you more with the detection methods. I hope this all are textbook content, and already the previous speaker has been described about it yesterday, I believe. So to just highlight, there are at least 2 different techniques, but there are multiple techniques to detect copied the number of DNAs in the PCR machine. Most commonly used is the cybergreen and the tagman. So cybergreen is a dye which can able to go and intercalate with the double-stranded DNA. So whenever the DNA is copied, after making a single copy, it will be double-stranded. After making each copy, it will be double-stranded. So cybergreen will go and bind to the double-stranded DNA, and it is fluorescent molecule so that it has a different excitation and emission wave. When it was excited to a 480 wavelength of 480 nanometer, then it will give an emission of 520 nanometer, and this will be going to be detected as a fluorescent signal. And whichever you are seeing as a peak or a graph in your real-time PCR is just because of the outcome of this excitation wave. Similar to the tagman, tagman has the fluorescent molecule tagged with the primer probe. So whenever the tag polymerase is active, we'll just cut this fluorescent probe and release, and so that we will be getting the fluorescent signal. So these are the detection methods in the cybergreen and the tagman are the detection methods in the polymerase chain reaction. Yes, now we know about polymerase chain reaction. It's a simple thermal cycling process, so it can able to amplify the DNA. So what's the problem with the DNA? So can we go and amplify any set of DNA, anything that we wanted to amplify, anything we wanted to identify? So how do you identify and detect a specific sequence in the genome? This is the question that's going to arise now. Because in the PCR, if you see, we are just going to amplify a gene or a segment of a gene, or a short fraction of the gene. We are not going to amplify the whole genome. So if you want to amplify a short sequence within of a gene, so there is 2 big issues we need to address. One is the specificity, another is the amplification. So what is specificity? As I said, the chromosome is a larger molecule. In this larger molecule, we are going to identify only a short sequence. So whichever primers we are going to add in the PCR must go and specifically bind to that region to amplify that gene of interest. The second thing is amplification. The amount of DNA in the sample. Say I have already addressed all the issues. I have designed a primer that's specific to the region that I'm going to amplify. But if the number of copies is very lower in the starting sample, and even though the PCR is going to amplify, if it reaches a point where the signal is not detectable by the machine, the fluorescence is not sufficient enough, again we are not going to see the amplification. So the 2 main criteria we need to address in the PCR is specificity and amplification. As I said, how do we identify and detect a specific sequence in the genome? Think about the human genome. We are doing many studies in human gene expression studies, especially the PCR. Think about the human genome. The whole length of the human genome is 3.4 billion base pairs. Mostly your PCR will detect 1500 base pairs if it is semi-quantitative PCR. And if it is real-time PCR, the advisable product length is 70 to 150 base pairs. So from the 3.4 billion base pair, you are going to specifically amplify or specifically detect some 150 base pair region. How you are going to be more specific over that? Take a common housefly, it has 900 million base pairs. The rice, which you are eating every day, it has 400 million base pairs. The E. coli, the common bacterium, it has 4.6 million base pairs. And the virus that's threatening us today, the SARS-CoV-2, has the genome of 30 kilobase. So how we are going to specifically detect it? Taking for example, human genome is 3.4 billion, right? So if we extend our chromosomes of this 3.4 billion, it will extend to up to 5,366 miles. Just imagine the distance. If you can't imagine, I'll just give an example. The distance between Kashmir to Kanyakumari maybe comes around 2,200 miles. So which means our human chromosomes will extend at least 2.5 times larger than the distance between Kashmir to Kanyakumari. So in this, we are going to identify 500 base pair segment. So which is almost like 4 feet. Maybe double the size of this, it's only 4 feet. So how we are going to identify only this much segment in the larger chromosome? So there lies the specificity. Second thing is amplicon to be detectable. Either in the case of semi-quantitative PCR, where you run the PCR and then you run an agar to detect it, or it's going to be a real-time PCR. You need to get a certain amount of concentration to be detected. Otherwise, it will go undetected. So with the starting material, that's why in the PCR we are amplifying the gene. That's why we are not just detecting. You may be having a question: if the gene of interest is already there in the sample, why can't I already take the sample, put it some dye, then I can visualize? That comes because of the concentration to be detectable. That's why in the PCR we are amplifying the product. Whichever we are getting some gene or RNA, we are amplifying to multiple copies. Say for example here, starting from 100 cells, we will be amplifying the DNA up to 180 million times in our PCR. It runs to 40 cycles. We will be amplifying from thousands to millions so that we can able to detectable and we can easily interpret the results error-free. And of course, as I said, there is two problems: specificity and amplification. And what is the problem for the solution? Is your PCR. Because PCR is the specific primer is going to detect the specific region in your gene of interest, and it is going to amplify that segment to multiple times so that the copies from thousands is going to copies from million so that you can able to easily detect that. So both the problem of specificity and amplification is going to be solved by the Pcr polymerase chain reaction. So Pcr reaction components. One sick thing that I'm seeing in the now upcoming generations is now as the technology has been improved, now for doing the PCR, mostly what people are doing is they are getting the Pcr master mix and then just they are adding with the RNA or cDNA they have. They doesn't realize what are the components in that. Previously, where the technology is less, we used to get each and every component separately, then we need to mix it together. I'm not blaming the technology. Technology is always good, but we need to understand why the technology is there, what are the components are there. So whenever somebody is performing in PCR, usually nowadays they are just getting a Pcr master mix, and they are mixing this Pcr master mix to get the DNA amplified. So in our classical system, the Pcr master mix will have, or the Pcr components will be having a water, buffer, DNA template, primer, nucleotides, magnesium ions, and DNA polymerase. These are the essential components to be there in your Pcr to perform a reaction, and each one of them have a specific role. And water is just going to act as a medium to make up the volume similar to each the vials. So buffer is the one that is going to stabilize the DNA polymerase, because DNA polymerase is an enzyme, as you all know. Each and every enzymes need a specific pH to be active, and specific temperature to be active. That's why chemists is doing extension at a higher temperature of 80, because they can perform only at that temperature. So the buffer is the one need to give the essential required pH, and DNA template is the must, because we are going to amplify only the DNA template. The DNA template can be your cDNA template, or the whole genome, sample from the genome, or the DNA which has been arrived by reverse transcriptation from mRNA to cDNA. Anything can be, but DNA template is the must, because in the PCR we are just going to amplify the DNA template. So we have the DNA template, but still to amplify the specific region in the DNA template, we need to have primers, that's forward and reverse primers. and that primers must be specific to the regions of within the gene of interest that we intended to amplify. Usually there will be two primers, the forward and reverse primer, and mostly the length of the primers will be 15 to 30 nucleotides. And after that, we'll be having nucleotides in the PCR reaction, because when we say that the gene is amplified, means we are adding ATGC, we are adding the genes. While the DNA polymerase synthesis new stand, it's going to add these nucleotides, A, adenine, thymine, guanine, cytosine. So without the nucleotides, the PCR reaction is not going to happen. So in the mix, there will be nucleotides, and there will be magnesium ions. As we know, the concept of cofactor and coenzymes, the magnesium ions will act as a cofactor for the DNA polymerase, so that the magnesium ions will activate and keep the DNA polymerase active. Without this cofactor, the DNA polymerase will be inactive or the activity will be reduced. And DNA polymerase is the master who is doing the action. As I said, there are typical compositions. This was just an example, and this may be vary from what type of PCR you are doing. And sometimes we'll be preparing a master mix, then we'll be separating the different vials. So most probably the TCHCl, the buffer will be in 20 millimolar, magnesium chloride 2.5 millimolar, potassium chloride 25 millimolar, TNTPs, that's nucleotides, of 50 millimolar. Then there will be two primers, forward and reverse, which will be in 20 picomole concentration each, and Jack polymerase, which is 2.5 units, and the template DNA, which can vary from 10 to 100 nanogram. Is it what the PCR reaction is doing is like, it's just unwinding it at the initial denaturation. Then after that, your Taq polymerase, the primers is going to bind. After binding of the primer, your Taq polymerase is going to come into action and to extend the strand to make multiple copies of the DNA. So while we are starting with the short complete DNA, at the end we are ending up with the multiple copies of the double-stranded DNA. So to understand and make you more comfortable with what we know better, so we all know RT-PCR is one of the detection methods for SARS-CoV-2. So the PCR method has been applied in different aspects, whether you want to know only the presence of the gene, or you want to know the expression of the gene compared to some other gene, or to compare the expression of the gene in two different conditions, or to identify or to screen the pathogens as a diagnostic tool. So depend on that, you need to validate the method. So here, for example, if you see the method validation for the detection of SARS-CoV-2, that's done in a very meticulous way. If you see the PCR reaction, how it was done for the detection of SARS-CoV-2, usually the sample has been taken from either from the oral swab or the nasal swab. After that, because it's expected that the swab will contain the viral RNA, SARS-CoV-2 RNA, then the RNA primer will be added to it to convert this viral RNA to a cDNA. Then to this cDNA, sequence-specific primers will be added to amplify. So if you see how meticulously this experiment was planned and conducted, there will be two set of primers in the SARS-CoV-2 detection PCR test. There will be at least two set of primers. That is for nucleocapsid 1, N1 and N2. So SARS-CoV-2 has like nucleocapsid protein, S protein. So in the PCR method for SARS-CoV-2, there are at least two primers specific to SARS-CoV-2 has been used there, N1 and N2. Then one primer is like, which can able to universally detect many coronavirus. Why I say universally detect many coronavirus? If you think that SARS-CoV-2, the coronavirus is new to the world, that's going to be wrong. The coronavirus is existing with us from at least from 1973, when it was first identified in UK. But it was an infection of pneumonia or something like that. From that, we have different coronavirus in the different forms. Even our earlier infections, like SARS and MERS, are caused by a type of coronavirus. So there are this coronavirus, this primer is conserved between different coronavirus, not specific to SARS-CoV-2. And at least it will have one primer that's specific to the human gene. Why it should be specific to the human gene? And if you have a non-template control, which should not express, and a positive control which should express. So after taking the sample and taking reverse transcription and doing a primer, doing the PCR with two primers specific to SARS-CoV-2, one primer specific to general coronavirus, and one primer specific to the human gene, not related to the virus. The end result will be given positive or negative based on the result. I said CT values. Okay, one thing. And say, for example, in a sample, if the both primers for both SARS-CoV-2 N1 and N2 is positive, and also N3 is positive, and RNA-sp, this is to the human. These three are related to the primers are related to the virus, and this one primer is related to the human. If all are positive, then the result will be given as positive. Consider if either one is positive, also that's considered as positive. But say if both are negative, that's a primer specific for the SARS-CoV-2, the coronavirus giving negative, but still the primer that's specific for the conserved sequence of the general coronavirus is positive, still it will be presumed positive, means we need to repeat the test after some time. In such a case, where like related to specific to SARS-CoV-2 is negative and the general for coronavirus is also negative, but when your RNA-spe that's related to the human is positive, then only the result is given as negative. If all are negative, then the test will consider as invalid. This is one of the major reasons why we want to have internal controls in our detection methods from the PCR. This is, RNA-spe is going to act as an internal control. Why? Because a technician is taking a swab. Considering that there is a SARS-CoV-2, suspected to be SARS-CoV-2. So after taking the results, if everything is negative, we are not sure whether the swabbing is proper. Whether the virus has been picked up from the nose or the throat, we are not sure. Or whether there is some technical error which has not amplified any of the genes. So that's why the negative will be given only if the RNA-spe related to the human is positive. What it means is the swabbing is proper because we have scrapped the tissue so that we can able to get the cells or the gene from the human tissue, either from the nose or the throat, but still it's negative for virus related thing, then it will be given as negative test. Okay. Coming back to our PCR related things with our aquatic thing. Just thinking back after years, I was just wondering when I get the invite from university, why from marine industry, marine science is inviting me. Then just recalling my PhD days, I just realized that my PhD is entirely about the aquatic thing. Being a basics in biochemistry, and of course, done fusing some microbiology and involving molecular biology techniques. In my PhD, I was working with two coral bleaching pathogens, that's called Vibrio chiloni and Vibrio coraliticus. And of course, as you all know, coral bleaching is one of the marine diseases which is disturbing the ecological system. Coral is an animal which can able to form the calcium carbonate skeletal, which can live in groups of the polyps. And usually the corals are beautifully colored because of the presence of algae. The algae and the coral animal is in a symbiotic relationship. So once either the algae dies or the animal dies, so the symbiotic relationship is lost, leaving behind the bleaching pathogens. So prior to that point of identifying the coral bleaching pathogens, it was believed that the other environmental factors that's causing the coral bleaching, maybe like sedimentation, which is not permitting the light to enter in, so that the algae is not growing and it's disturbing the health of the animal also. But there is a point when they discovered there are few bacterial pathogens which can able to infect the corals, leading to the coral bleaching. And the one important thing we need to note at that point is these bacteriums are pathogenic only at higher temperatures. So only in summer the pathogenic determinants are getting expressed and it's going to lead the coral bleaching. On other days, in the winter days, nothing is going to happen. So our PhD work, our research work starts from this point. So if only higher temperature is triggering the pathogenesis, triggering the coral bleaching, which means there is some factor within this bacteria which is getting expressed in higher temperature and which is not going to get expressed at the lower temperature. So we went to identify what are those factors. And burden apologize me to having the semi-quantitative PCR results. Please take it from me. At that time, these are the highest technology which we could afford and which is available. Not the real-time PCRs are not classical at that time. So at least we have thought there are three different genes: Toxar, Toxes, and OMPU. We hypothesized that these genes may be the virulence factors of this bacterium, and this may be triggered the expression of higher temperature. So to prove this, we have taken the help of the PCR. So what we have done is we have grown the bacterium at two different temperatures, 23 degree and 30 degree temperature. We have used Vibrio and Vibrio coraliticus, and we have grown it at two different temperatures. One is the non-pathogenic temperature, less than 30 degrees, that's 23, and one is the pathogenic temperature, that's 30. And interestingly, we found that all these genes which were expected is with a higher expression. The lower temperature, higher temperature, lower temperature, higher temperature, lower, higher, lower, higher, lower, higher. So you can see the bandwidth. So we observe that at the higher temperature, all these genes are getting expressed. Means these may play a role in the pathogenesis. So this is how we utilize the technology of PCR to identify the pathogenic response of the bacterium. What are the factors that can able to express higher temperature, which can lead to the pathogenesis. But and of course, after expressing all these genes, Toxar, Toxes, and OMPU, we went to sequence those gene products, and we have submitted the gene product to the nucleotide GenPack nucleotide sequence of the PubMed. It's in the direct submission. So why that sequence is very valid for us at that time is because now if you think whomsoever has already done a PCR, know that they need to use a specific primer to do the PCR. So how they get the primers? Most probably they will take the primers from the previously published primer sequence, or else they will be having a gene sequence, and with the gene sequence there is an option in the NCBI primer blast or primer tree, and also there are many other primer designing tools are available. If you have the accession number or the sequence of the whole gene, you can give that sequence to that software, and in turn it will give you a prime forward and primer reverse forward and reverse primer sequence. But while we hypothesis that Toxar, Toxes, and OMPU can be the pathogenic virulence pathogen determinants at higher temperature, and want to look for the expression of those genes, we doesn't have the sequence for those genes in the Vibrio Shilla, Vibrio Coriolis, and Vibrio Mediterranean. So what we have done is we have took the help of bioinformatics tools. On this day, I will remember and recall my good friend Mr. Anbaragan, and who has prompted me about the techniques of bioinformatics tools, where you can identify the primer sequence using a closest relative. What is the closest relative? If you see in the congested place here, this is about the phylogenetic tree. So what is a phylogenetic tree? The phylogenetic tree will be derived. It's a bioinformatics tool. The phylogenetic tree will be derived. You say, for example, on the 16S RDNA sequence, it is supposed to be the conserved sequence with the related species. So this will give you an idea of the closest relatives of whichever the new bacterium you are looking into. For example, take it in my case, I am looking for Vibrio Shilla. Okay, I don't know the gene sequence for Toxar, Toxes, and OMPU. But what I can do is I can look for the closest relative if the 16S RDNA sequence is available. So taking the closest relative here, then there is a bioinformatics tool called as cluster W. That's a multiple sequence alignment. So after taking the gene sequence, available sequence from the closest relative, because I was looking for Toxar, Toxes, and OMPU, for which there is no gene sequence available for the bacterium at that time. Now it's available. Now the whole genome sequence itself is available. So since I don't have the gene sequence, I cannot be able to go and design a primer for the gene. So what we did is we analyze the closest relative, then we pick the gene sequence for the closest relative, then we put it into a bioinformatics tool that's called cluster W. So cluster W will align the genes from the closest relative, and it will say which are the regions that is highly conserved between the different bacteria. So we assume that since these segments are highly conserved, means there is no change in the sequence between, say, for example, strain 1 to 1, 3. I'm looking for strain 4. So we assume that, so since this is conserved in the closest relative, the same region will be conserved in the unknown gene of interest. So we designed a primer for the conserved region, and then we amplified. This is the amplified product. And then we sequenced it, and then we have submitted to the GenPack nucleotide submission. And these are the 9 nucleotide sequence that's available in the GenPack related to Vibrio Mediterranean, Vibrio Shilla, and Vibrio Coriolis of Toxar, Toxes, and OMPU. That's from us. And this is how it looked like if you open each and everything. Not only for this, for any nucleotide sequence, if you want to trace from the PubMed, you can go to the GenPack. You can give the accession number if you know. Otherwise, you can just search the name of the gene. Say, for example, you are looking for spike protein of SARS-CoV-2. You can just use a spike SARS-CoV-2. You will get the gene sequence. So this is how the data will appear, and you can see for submission author's name, including me, Dr. Badiraja, and our guide, the mentor, Professor Murali. And of course, this was submitted on 18 May 2008. So whichever gene sequence you are searching in the nucleotide base pair, it will give a sequence like this. So you need to have this gene sequence to design a primer. If this is available, you are lucky. You just take the gene sequence, use some primer designing tool, you will get the primer. You get the after getting the primer, you synthesize the primer. You run a PCR using the primer. You can amplify that particular gene. Only if you are unlucky or if you are really hitting hard to find a new gene, then you need to go and analyze your phylogenetic tree. You need to use the cluster W. You need to use the blast to arrive at a decision of which sequence I need to pick for the development of my own primers. Okay, that's the history while I was coming with aquatic science in my PhD. So I will just give a few more examples where you can able to apply the PCR, because PCR is a technique, right? PCR is a technique, just a technique. As I said previously, it was known as the scientist tool which is used for the gene expression. Now even a common man know that it's a diagnostic tool for COVID-19 because it has various applications. So we just highlight few applications, examples where the PCR has been already reported or published to be useful in determining the aquatic animal health. Here is a study in 2010. It reports about the use of microalgae. Again, when I speak about microalgae, I cannot miss remembering my friend, my brother, Dr. Mayakannan, who is an expert in the microalgae. Of course, Dr. Mayakannan has been propagating the microalgae in various things. But here, the microalgae, what the authors did is they have taken the microalgae and they have transformed few vectors into the microalgae, and they have checked the efficiency of the microalgae to pick up these vectors, the viral vectors or something plasmid vectors. Okay. Why this study is more important? So here, why PCR is important for this study? So as they decided to transform the plasmid vector through different techniques, maybe the electroporation, glass beads, and PET media. So once they transform the viral vector, see, they have transformed the vector through different modes. But to analyze and identify whether the microalgae has taken up the electroporation technique, whichever technique to transfer the plasmid is successful, they need to extract the DNA back from the microalgae and then do a PCR to confirm that the plasmid is already there. So that's why it was done. So the vector they have used is like two different vectors they have used, the authors, the PGMT and PUC19. So if the vector is taken up by the cells, then if you do a PCR specific to any of the sequence in this vector, then you need to get the response for the PCR. So that's why they are able to get the response for the particular plasmid. You are getting my point, right? There is a microalgae. Now the author's decision is to transform a plasmid to the microalgae. They use different techniques. But how can I go and identify the success of that transformation? Whether the plasmid is already transformed inside the microalgae. Now I need to extract the DNA or RNA from the microalgae. Then I need to PCR specific to the regions within the plasmid. If that is going to get amplified, then I can say that the plasmid is already transformed to the algae. And of course, they went one step ahead to go on sequence, which is not essential actually, but they have also sequenced and proved that the gene of interest is already there within the microalgae. They have successfully transformed uh, uh, the vector into the microalgae, and they have shown that the electroporation is the best technique. Okay, now we have shown that PCR technique will be helpful to check whether my plasmid is transformed into a microalgae. Where I can apply this technique. Okay. One step ahead, the same authors, the next year, they came back and they used the tool to vaccinate the animal from vibriosis. As you know, vibriosis is the pathogenic vibrio going to affect the fish and leading to pathological sequences. So by successfully transforming a vector, now the author has introduced in the vector, the author has introduced various fragments of the OMPU that's going to act as an antigenic protein to create the immunization in the fish. So what they did is they have made a different constructs containing different fragments of the OMPU from the vibrio, pathogenic vibrio, and they have used different vectors to transform these vectors to the microalgae. Why this study is going to be important? As we all know from the aquatic background, from the marine biotechnological background, you know that microalgae are widely used to feed the fishes, the farms, because that gives more nutrients. And even there are already technologies existing where they are transforming a vector which can able to express the growth factors in the microalgae so that when the growth factor is already expressed, whichever the fish or the aquatic animals eating this microalgae will get more amount of growth factors, go higher numbers. Similar technology they want to adapt to vaccinate the animal. So what they have done is they have made a different construct using different fragments of the OMPU, that's outer membrane protein of the pathogenic Vibrio. So different fragments, they just cloned it into the vector and they put it back into the microalgae and they propagated this microalgae. But while propagating this microalgae, we need to get an idea for how long the gene will be expressed in the microalgae. To confirm that, they have grown from different generations like F1, F2, F3, F4, F5, F6. And in the different generation, they have isolated the DNA and the RNA and they look for the expression of this particular fragment, whether it's there or not. So looking into that, first they want to confirm the transformation efficacy. So they transform using the technology of homologous recombination. I don't think I need to go and explain you the homologous recombination, right? Because that's a separate chapter to discuss. But if you want to say in a simple way, what is homologous recombination? There will be a specific gene, there will be gene in the chromosome. Say if the gene is defective or if you want to change the gene, during the using this homologous recombination, since the homology of the sequence, in the homologous recombination, we will use the same sequence, the complementary sequence, to design a vector. So based on the homology, the vector will just go and replace the portion in your gene because of the homology. It's called homologous recombination. Here, the homologous recombination they are used as a knockout. Means like there's already a gene made a complementary the vector was made complementary to carry the homologous region of this gene and also containing the different fragments of the OMPU. So due to this homologous recombination, this vector will go and just replace the existing gene and it will sit inside there. So to confirm that, the actual size of the gene which they have replaced is 980 base pair. So wild type, whichever has not taken the plasmid, will express that at the 980 base pair. That is confirmed by the semi-quantitative PCR. Through the homologous recombination occurred successfully, if the gene was replaced, then this vector has been sitting inside the host chromosome. Then it will take the size of 3703 and 4208 base pairs, depending on the two different vectors. They use PCP OMP ZA1 or VCP OMPK ZA2. There are two different ones. So to confirm the successful transformation and integration of this vector into the host chromosome, again the PCR will be helpful to detect if you know the sequence. And also the PCR is again helpful. Okay, now see the result of the first paper. They have through the PCR they have confirmed that the plasmid can be transplanted into the microalgae. They went one step ahead to confirm whether this can able to integrate into the host chromosome and the particular segment fraction of the OMPU, the OMPW from the pathogenic nuclear species can able to get expressed in the microalgae. So there's screen for different generation, and they have found that up to different generation, at least up to F4, fourth generation, they can able to express the whichever microalgae carrying the fragments of this OMPU can able to express the genes. If they can able to express the gene, they can able to produce the antigens. You know the concept of antigen, right? In the concept of vaccine. The concept of vaccine, antigen will be given, an antibody will be raised against the antigen, so that whenever the real virus is coming to attack, since our body knows how to act to the antigen, it will act immediately, respond immediately, and it will clear off the infection. Similarly, to treat the vibriosis infection in the fish, vibrio infection in the fish, to vaccinate the fish, the microalgal technology has been used. In the microalgae, they express the antigenic determinants of that particular pathogenic bacterium. They express in the microalgae. So once it is expressed, the microalgae will contain an antigen, something it will act as a oral vaccine. Even if you consider for the humans, the polio vaccine is the oral polio vaccine. So when the animals or the aquatic animals are feeding this microalgae containing this antigen, then the animals will be immunized. Why we want to do the generation studies, how long it will survive? Say for example, now we know that we can able to successfully transform the plasmid containing the antigenic determinants, and we can able to produce the antibodies inside that. We are propagating the microalgae in the culture or in the laboratory to feed to the animal. So if we go beyond F5, F6, X1, 8 generation, we are seeing that already it's lost. The expression of these antigenic proteins are lost, antigenic genes are lost. So this will give you an idea. Even if you want to use the microalgae to vaccinate and to prevent the aquatic animals from the vibrio infection, acting as a source of antigen, you can able to propagate only up to which extent, which generation you can able to propagate the microalgae to get a successful vaccination. Of course, this is another example where you can use the PCR technique to screen for the antigenic determinant if you want to use the antigen to be expressed as a oral vaccine. And here, one step ahead, they went, okay, we know how to transform a plasmid. We can screen through PCR. After transforming a plasmid, we can screen whether it's integrated into the host chromosome. After integrating the host chromosome, we can screen for the particular gene and we can confirm that whether the particular gene is expressed. Now done. Okay. Consider just in the concept of vaccine. Now we have done a microalgae, everything. Now we have given it to the animal. Animal has taken it. How do you confirm? How you can use your PCR technique to confirm that the animals are getting immunized? Your animals are getting immunized. Only if the animals are getting immunized, then it can act. It will overcome the real infection when it's coming. So what has been done is through the PCR technique, we can go and analyze the expression of the genes that are responsible for the antigen recognition. So here in this case, we see there are different tissues has been analyzed like kidney and spleen, and different antigenic determinants has been given in a different study. Here you can see that OMPK has triggered most of the antigen recognition related genes in both the tissues. So what it means is, comparing these two antigenic determinants, OMPK can act as a better antigenic determinant so that it can able to trigger more antigen related genes within the host, within the aquatic animal, so that the animal can get immunized. So with the PCR technique, you can even able to go on screen which portion of or which component of the pathogenic strain can act as a better antigenic determinant so that the animal can be better immunized. And again, this is another example where the lactobacillus casei containing the flap B has been given. And again, it was seen like how the antigen responsive genes has been expressed to the PCR. So through the PCR, you can screen everything. We will usually say that PCR is just a gene expression technique. Yes, PCR is just a gene expression technique, but it depends on where you are applying, how you are applying, what you are detecting. So it will produce more meanings when you think where you can able to apply and how you can able to get the results and how you are going to interpret the results. So what we have seen just now in the aquatic animals, in the aquatic health, PCR will let us know to decide on the gene manipulation strategies, how we can manipulate the gene, how you can put the gene into the vector, how the gene can be expressed, whether the gene is expressed or not. So PCR will tell us to know whether the gene of interest is transferred to the host. PCR will let us know whether our gene of interest is already expressed or not. PCR will let us know what are the molecular changes happening within the cell by the expression of this particular gene. Okay. So aquatic health, PCR is just a gene expression technique. But through this gene expression, you can able to decide whether your gene of interest, which you want to express in a different animal, is getting transformed, integrated in the host chromosome, getting expressed, and what are the after it's getting expressed, what are the changes it can induce to the host chromosome. All these changes we can able to screen or monitor through the polymerase chain reaction. Coming to the to make it very understandable and very easy for you to understand, whatever the vaccine concept we are discussing for the animal. Just think about the vaccine, which is commonly known as Covishield now. Actually, the name which was given to the Covishield, the SARS-CoV-2 vaccine, which many people are taking now, is a Covaxin Covishield. The initial name given to Covishield in the developmental stages is called chimpanzee CHADOX1. What it means is chimpanzee adenoviral vector developed by the Oxford University for the novel coronavirus 19. That's the name before SARS-CoV-2. It was called as nCoV-19. The vaccine concept, which I have explained for the fishes, is almost similar to the concept of how the vaccine was developed for SARS-CoV-2 in the Covishield. So what they have done is they have taken a chimpanzee adenovirus and they have modified to the replicated deficient. Means this is an adenovirus which can able to infect the monkeys, not the humans, and it was modified to be non-replicative. To administer the live virus, the virus will go into the host but it will not replicate to the host. Okay. Then what has been done is, we already know the whole genome sequence of the SARS-CoV-2. Either we can extract from the genome and cut it the gene for the spike protein, or we can do a PCR to amplify the region for the spike protein, or we can synthesize the gene for the spike protein. And the gene segment that is responsible to produce the spike protein in the SARS-CoV-2 has been administered into the adenoviral vector. Now think where the PCR will help. Like similar to the examples of the vaccine I have given to fish. Here, if you want to know that you are getting the exact portion of the spike protein of the SARS-CoV-2, you need to have the specific primers which will be going to design based on the whole genome sequence of SARS-CoV-2. So here your PCR will help to amplify that particular region. Otherwise, like every time you need to grow a virus, cut the specific region, put it back. So you need to propagate virus in a larger quantity, which is going to be risky. So as you already know how you can able to amplify that particular segment using the PCR technique. You can extract the DNA, you can extract the RNA from the SARS-CoV-2, do a reverse transcription, do a polymerase chain reaction for the particular spike protein. So the PCR will give you multiple copies of the spike protein, the genes for the spike protein. Take that and put it back into the adenovirus. Now, through using the PCR, you amplify the spike protein region, you have transformed it, you have put it back to the chimpanzee adenovirus. How do you know that the chimpanzee adenovirus is already taken up the spike protein? For that, again, you need to go and do a PCR, screening PCR for the same region, and you need to confirm that whichever the chimpanzee adenovirus is there in your culture, which you want to go to administer as a vaccine, is containing that specific region. So after that, it will be given to humans. So once this replicative deficient chimpanzee adenovirus containing the spike protein of SARS-CoV-2, that's called Covishield simply, okay? Covishield is, otherwise technically we can explain this. The replicative deficient chimpanzee adenovirus containing the gene to express SARS-CoV-2, the spike protein of the SARS-CoV-2, the Covishield, when it's administered to the humans, after going to the human body, it's going to trigger some immune response. So what kind of immune response that's going to trigger, what kind of immune cells that's going to trigger, what kind of downstream signal that's going to be activated, all these things you can able to screen by PCR. So I said PCR is just a gene expression technique, but even the simple example of both in the fish and in the Covishield, you think that PCR will be helpful to recognize a particular sequence in the gene. PCR will be helpful to amplify a particular gene. PCR, after amplifying, after constructing a vector, putting into some post, PCR will be helpful to screen the positiveness or screen the effectiveness or screen the presence of the particular gene in the virus. PCR will be helpful. After putting it into the humans, even the immunological response can able to go on screen by the gene expression studies. So even though gene expression, PCR has applications at multiple angles and multiple phases. Okay, quickly, I will just give an example how PCR applications can be used in aquatic toxicology. Just giving an examples from the published report. Say, for example, there is an endocrine disruptor. There is a chemical, or you want to screen a new chemical, and you want to know whether it can able to go and disrupt the endocrine regulators or not. So already in a study where they used estradiol treated with estrogenic compounds, and they have analyzed the sequence of genes that can able to respond to this estrogenic compound. And in this study, they have concluded that the more sensitive nature of ZR, that is zona radiata, suggested that ZR gene is more suitable indicator that we can give to estrogenic substances. So here you can see this gene, the ZR gene is giving statistically higher response to the estrogenic compound. So if a compound is estrogenic, then adding that particular compound to that particular fish is going to activate this kind of signals, which we can able to screen by the real-time PCR. If this is not going to get activated, then you can say that whichever compound you are adding to the culture or to the fish is not going to be estrogenic. So you can screen which are the compounds which are estrogenic. So if you know that if a particular signaling is get activated, and you can able to analyze the gene expression of that particular signaling cascade with PCR, then if this signaling cascade is going to be disturbed, that also you can able to analyze with the PCR. If it's going to be activated, the gene is going to be overexpressed. If it's not going to be expressed, the gene is going to be downregulated. So with this idea, you can able to get an idea whether the compound is going to be an endocrine disrupting chemical or it's an endocrine analog or endocrine. So similarly, to identify the toxicity of the metals. So here what has been done is the zebrafish was exposed to different metals like copper, cadmium, mercury, and zinc. And they have analyzed the expression of the zebrafish metallothionein, this particular enzyme, by PCR. So they have seen that the expression of the zebra protein metallothionein ZMT gene is higher when they exposed to these metals. So which means whenever the metal is at the toxic concentration, whenever the metal is toxic, you can see the higher expression of ZMT. So what you can do in an aquatic toxicology study is you can get a zebrafish, treat it with anything you expect or you presume or you know that it has a heavy metal. And if you see that there is a higher expression of ZMT gene by the PCR, you can conclude that these metals are toxic to the aquatic system. If the ZMT is not going to be expressed highly, then you can conclude that whatever the specimen or the sample you are adding is not going to be toxic to the aquatic environment. So to screen the toxicity of the chemical or the compounds just by the gene expression, your PCR will be helpful to do that. And also to screen whether the pesticides are harmful to that. Similar to the example which we have just seen for the metals, if you already know an idea how these pesticides will work, or expression of certain set of genes is going to suppress the growth of an aquatic animal. See, if you say that certain, you already know that there are certain set of genes that need to be expressed in the developmental stages of your aquatic animal. And adding these compounds is going to suppress those genes, means it's going to suppress the development. So just by seeing the expression or suppression of the gene, you can conclude that whether the pesticides or herbicides that we are using nowadays, whether it's going to be toxic to your aquatic animal. So PCR is gene expression, yes, PCR is gene expression technique, but you can apply it into different aspects. So what we have seen, the application of PCR in aquatic toxicology. So PCR will let us know to understand the toxicity of the compounds. PCR will let us know on the biomarkers that need to be analyzed during the toxicity study. So PCR can act as a biomarker. Say for example, if already a known toxic compound is there, and if the known toxic compound has been added to the aquatic culture and certain set of genes have been expressed, and if you already identified that if the compound is toxic, this particular gene will get expressed, then you can fix that particular gene as a biomarker. So next you have an unknown compound where you test the toxicity and you are adding that to the aquatic animal. While you are adding this, if that particular biomarker gene is going to get expressed, you can simply conclude that this is a toxic compound. And if the particular biomarker is not going to be expressed in your PCR reaction, in your mixture, then you can conclude that this particular compound is not going to be toxic to the aquatic environment. And also PCR will let us know on the affected tissue. If you take the tissues, different spleen, kidney, brain, and analyze the expression patterns of the different tissues by PCR, it will tell you which organ is get more affected by the toxic animal. Okay. So we know how the gene expression technique, the PCR, can be applied to the aquatic health and aquatic toxicology. But whether PCR is helpful in predicting the disease outbreak, because what all we have seen is we can prevent the disease using the PCR technique. We can identify the toxicant. Whether PCR can able to predict the outbreak, because now there is a pandemic outbreak, SARS-CoV-2. We all are suffering. We are all in the same home. Just by technology, we are getting connected. So whether PCR will be helpful in predicting the disease outbreak. Just giving an example of the study which has been published in the year 2017. The study is about taking the samples from a pond, from a lake. Okay, taking the samples from the lake. They have taken different samples. I believe like the sand, any aquatic plants there, algae, fishes. From all these, they have extracted the RNA, and they have identified which sequence is conserved in pathogenic and non-pathogenic bacteria. They have isolated different Vibrio species in the lake from different from the soil, from the aquatic animal, from the plants, from the algae. Whichever the source is there in the lake, they have extracted the RNA and they have amplified it with PCR, and they have seen that there are few certain regions which is conserved in the pathogenic bacteria, which is already proven. Say, for example, Vibrio hobby. So the pathogenic sequence is already confirmed, and after analyzing nearly 300 isolates, they have seen that the conserved region in the pathogen is already there. In many of the strains, they have been isolated there. So which indicates that the pathogenic strains are already lying there for a longer time. Sorry, I just mixed with the studies. Okay, this is the study which can able to confirm you about the outbreak. What they have done is they have taken the samples from different occurrence of toxigenic Vibrio cholerae during a non-outbreak period. They have taken the samples in the 2017. Of course, the outbreak of cholera, Vibrio cholerae was happened in that region in the year of 1993. So what they have done is they have taken almost 360 samples from the plankton, water, um, the fishes, all these things. They have collected the samples and they have analyzed for the existence of the genes related to the pathogenic Vibrio cholerae. Because if there is the existence of the genes, assume that the Vibrio cholerae is already there in the water or in the aquatic region there. So by doing that, they have found that the study report that occurrence of multi-drug Vibrio cholerae in the lake, because they analyzed in the 2017, the last cholera outbreak at that region was 1993, which means the leftover of the outbreak, the cholera, the Vibrio cholerae outbreak, the cholera outbreak in the 1993 is still existing in the lake till the year of 2017, which means the pathogenic bacterium which has caused the cholera outbreak into 1993 is still existing at the region in the till the year 2017, but there's no outbreak yet. But through the PCR, they already confirmed that the bacterium, the virulent bacterium which has caused the outbreak in the 1993 is still existing at the lake of now the region. We don't know why the outbreak is not there. Maybe we can club with my own study where we can see that it is waiting for some environmental factor. At the lower temperature, it's not pathogenic, only at high temperature, it's not pathogenic. Maybe this Vibrio cholerae is waiting to see a favorable environment for it to getting expressed the pathogenic determinants so the outbreak can be done. So to screen the presence of this pathogenic bacterium, the real-time PCR technique has been used. So similar to that, if you see the current pandemic, the SARS-CoV-2, whether we can able to identify whether we can able to pre-identify the outbreak of any pathogenic determinants or SARS-CoV-2. This is study very interesting. If you see that how the SARS-CoV-2 or how the COVID-19 or SARS-CoV-2 can able to go and affect the aquatic animal. If we see that in the study, they have synthesized a peptide that can able to mimic a portion of the SARS-CoV-2 spike protein. As you all know, SARS-CoV-2 is the virus that's leading to the COVID-19, and spike protein is the one that's going to bind to the ACE receptor of the host and getting internalized, causing the major problem in the SARS-CoV-2 pandemic and the SARS-CoV-2 infection. So what these authors have done is they have synthesized a short segment of peptide that can able to resemble the spike protein of SARS-CoV-2, and they have added it to the cultures of the tadpole. So while adding it to the cultures of the tadpole, they have seen that through the in silico studies, they have seen that they have the interaction between the peptides and estrogen cholinesterase and antioxidant enzymes, and the aquatic particle contaminant SARS-CoV-2 can constitute additional environmental change. They have seen that the peptides of the SARS-CoV-2 is affecting the growth or the neuronal development of the tadpole. Till now, we have only assumed that the SARS-CoV-2 is affecting the humans. It's not going to happen. So we need to have an enormous study of how this pandemic is going to affect our aquatic life. Well, there is traces of this SARS-CoV-2 lying in the water, whether there is a traces of SARS-CoV-2 in aquatic animals. Because even recently we have seen the news that the lions are getting affected with SARS-CoV-2, and one lion has died because of infection of SARS-CoV-2. So the pandemic SARS-CoV-2 infection is not restricted only to the humans. So how you will identify the effects of SARS-CoV-2 in the aquatic environment? You can just go and screen for the presence of SARS-CoV-2 in the aquatic animals through the use of PCR technology. Okay, and in that screening, as I said in the Vibrio cholerae example, you need to identify between pathogenic and non-pathogenic. Similar to that, now we know that the double mutant features arise from the India, that's called as double mutant now, the Delta strain, Delta variant by WHO now, which is more pathogenic than the initial SARS-CoV-2. So whichever traces you are identifying in the aquatic environment, you need to design a specific primer that can able to identify the double mutant. It means you can able to confirm that whether you are having a highly virulent SARS-CoV-2 environment or less virulent SARS-CoV-2 environment. But however, all the strains are virulent to humans as of now. So to summarize again now about the study, this study they have synthesized a peptide that can able to mimic the spike protein of SARS-CoV-2. When they added to the aquatic culture and the tadpole culture, they have seen that it can able to create a neurotoxicity and the development of and inhibit with the development of the tadpoles. Okay, what's wonder there? How the aquatic animals are going to get infected by SARS-CoV-2? How they are going to expose to the SARS-CoV-2? You know, it's infecting only humans, right? Then you need to go and see the number of studies that's coming up. SARS-CoV-2 as detected in the wastewater. Because as we humans, we do many things. Even in the hospital waste, there is infected. There are many SARS-CoV-2 detected in the wastewater and in the sewage water. So as it was detected in the waste and sewage water that's coming out of the hospital or in the household things, all this water is going to the environment. Once it go to the environment, this is going to come and interact, come and closely interact with the aquatic environment. So that the aquatic environment is going to be get affected by somewhere or other with the SARS-CoV-2. Again, this is an example of another study where it says that the recreational waters, the potential transmission route for SARS-CoV-2. When they analyze the samples from the recreational waters, that's from the swimming pool and all, they can able to get the traces of the presence of SARS-CoV-2 in the waters, in the swimming pool, in the recreational center. Means the aquatic source, the aquatic environment is already contaminated by the SARS-CoV-2. And again, as we have just celebrated the World Environment Day yesterday, as we humans are always selfish, we care only about us. Even there is an improper disposal of the face mask that they are doing, right? We're just throwing it as such in the aquatic environment. Say, for example, a person who is infected with SARS-CoV-2 is using a face mask, and the face mask contains the droplets or the traces of the virus SARS-CoV-2, so we simply throw it in the environment. So, through some aquatic water sources, the SARS-CoV-2 is going to enter into the aquatic environment. So, what is the fate of the SARS-CoV-2 in the aquatic environment? How it's going to affect the fishes? How it's going to affect the crabs? How it's going to affect the phytoplanktons? We don't know yet. So we are concerned only about the humans, and we are making more studies only about the humans. So through the use of PCR technology at this pandemic situation, as a marine biotechnology student, you have enormous things to do and you have enormous things to discover about the effect of this pandemic in the aquatic environment. So for all these things, your simple PCR, the polymerase chain reaction, will be able to help you to go and detect whether the presence of SARS-CoV-2. You can start from your household, drinking water, the water in the pond near you. The whole genome sequence of SARS-CoV-2 is already available. So designing a primers for a specific region using the primer is not going to be a big concern for you. But even if you want to know more about the primer designing, that we can discuss later, because that's by itself a very big topic. So there are multiple projects which you can able to open up by the use of PCR. You can just go and screen the presence of PCR in the aquatic environment near you. Just the PCR. Just have a primer for SARS-CoV-2. Collect the sample, do a PCR. Collect the phytoplankton, collect the algae, collect the fish, collect the organs from the different animals, and just look for the presence of SARS-CoV-2. Then, if you already have a pre-existing data, how the gene expression will be in a normal animal, how the gene expression will be there now in the SARS-CoV-2. The animal contains SARS-CoV-2. If there is any difference in the gene expression, you can identify PCR. You can always tell what is the toxic effects of the SARS-CoV-2, what's the pathogenic effect of the SARS-CoV-2 to that aquatic animal. Again, it can reach the aquatic animals or the aquatic source in a different way, either the direct contact or indirect contact. So there are multiple potentials where the SARS-CoV-2 can able to reach the aquatic environment. So as a marine biotechnology student, you have larger number of projects that can be able to open up where you can use PCR to detect. So don't just assume PCR is just a gene expression technique. So PCR is a technique to be thought beyond just the gene expression. So now the SARS-CoV-2 COVID-19 has opened up research in many areas, including the aquatic health and toxicology. And PCR has enormous potential to answer all your research questions. Okay? So you have a lot of projects which we can do related to SARS-CoV-2 itself. You don't need to go and play with the virus. You don't need to go to the virus. Since the viral genome, the whole genome sequence of SARS-CoV-2 is already known, you can first start from screening the presence of SARS-CoV-2 in your aquatic environment. That will give an idea whether we are going to end this pandemic or we are going to live with this pandemic forever. Because once the aquatic environment is contaminated and that can act as a carrier for the SARS-CoV-2, there is no way or there is no chance that we are going to get eliminated out of this pandemic. So we humans need to think beyond selfish. We need to think that the world is for each and every animal living in this world. The world is for each and every life in this world, not only for humans. So we need to spread the virus. We need to stop the spread. We need to save the environment. So quickly, what you people can do is you can start up with multiple projects to screening the presence of SARS-CoV-2 in the environment, aquatic environment. What is the effect of SARS-CoV-2 in aquatic animals? So all this. By this, I will just conclude my session. Say thank you. And apologies me, I was looking for some old pictures. That's why I was looking for some old pictures to remember it. Of course, I don't know how many of you can able to recognize this simple and humble human being standing there. This was the photograph taken on the day of my YOC, 2008 PhD Viva. And this humble human being just residing on the glass door here is Dr. Jay Prakash Vel, who is supposed to be the star in charge of it. I'm sorry, Dr. Mayakannan, Dr. Muthalan, and Dr. Selvakumar. They are also somewhere around here, but I cannot able to trace my old photographs because although the technology is not higher to get a good quality pictures or many pictures. But anyway, I'm happy to be associated with all these people. I'm happy that we are associated nearly 2 decades, nearly 20 years, and our things will go on. And we'll be looking for more interactive discussions or interactive projects like that. So thank you. And at this point, I need to thank and acknowledge my professor, Dr. S. Murali, who has introduced me about PCR. As I said, PCR, I started my PhD as a semi-quantitative PCR. Later, even I have done real-time PCR using Blackman and cytokine. And of course, even I have done a multiplex PCR where multiple genes can be amplified in a single well. Because usually, even in a real-time PCR, you can amplify one gene per well. There are multiplexing technologies, multiplexing PCR available now, where you can amplify more than 80 genes in a single vial. And thanks to the organizers, like Dr. Prakash Vel, Mike Kannan, Dr. Muthalan, and whomsoever I'm not mentioning them, please bear with me as I don't know all the names. And thanks to the GLR laboratories, which is the current employer, who has given me generous permission. Of course, GLR is doing toxicology studies not related to aquatic, but they are doing the toxicology studies related to biomedical devices. Thank you all. I will stop my lecture here, and I will be happy to pick up any quick questions related to this.
Thank you, sir, for the wonderful lecture and giving us a detailed information about PCR. The session was really informative. Now we are open for interactive session. Participants can click on raise hand option or post questions in the comment box session.
I have a clarification, Dr. Murali.
Yes,
It is
sir.
with regard to, we are always aiming at the spike protein to neutralize the effect. Why don't you go for the body protein outer covering of the virus? Because sometimes the mutations can change the outer spike proteins.
Yes, sir. Actually, that's the problem happening now. Spike protein can able to easily mutate. Actually, even if you want to go more deeper, and actually your question is more valid about spike protein. No? The major difference between SARS and SARS-CoV-2 in the spike protein is the change in just 4 amino acid residues in the spike protein. Just the change in these 4 amino acid residues from the SARS-CoV-1 to SARS-CoV-2 made the virus highly virulent, highly propagating, highly infectious, causing a pandemic. So each and every protein of that of the SARS-CoV-2 is highly important, including the spike protein and nucleic acid. Yes, sir. Your question is much valid. We need to analyze each and every protein of that, because as of now we don't have any idea on that. We are just thinking only the spike protein. Even the vaccine trials, if you see, to use as a vaccine candidate, they have tested different antigenic determinants of SARS-CoV-2. Even few studies says that nucleic acid can act more better than spike protein as antigenic determinant for the viral candidate. But up to my knowledge, no viral candidate has been developed for the nucleic acid protein of the SARS-CoV-2. They are focusing only on the spike protein. But as you say, your question is much valid. As you say, each and every surface protein on the protein of the nucleic acid of SARS-CoV-2 is much valid. Thank you, sir.
Thank you. Thank you. And another thing is, my clarification is, I think your lecture now, it gives information which is much more to be worried about.
Yes,
Contamination. It is something good which we need to make this information as awareness among the entire mass, because we should not indiscriminately throw away the contaminated things, and which will lead to, as you said, we may have to live longer with this pandemic virus. And this valid information need to be addressed through the media and social media to the common man. So
yes, sir,
really,
already many studies are coming up. They say that they can able to see the traces of SARS-CoV-2 in wastewater, treated water, and aquatic animals. But as of now, the reports say that it's existing there. But we don't know the consequence. What are the
yes,
damage is already done? How we can able to prevent the damage? Whether we can able to prevent the damage also, we don't know. Because we by ourselves cannot able to save us from the pandemic virus. We don't know how it has affected the environment. Even though if you consider that the aquatic animals are just carrying, not infected, but we don't know the fate. After that, even if you assume we are coming down with the pandemic, we don't know whether we'll be getting reinfected through the aquatic environment, the aquatic source.
yeah. As you said, even the lion in the tigers in the case of it is getting infected. It is not human to human now. Even it can go to some lower vertebrates. The multiplication will be much higher than in the homeothermic organisms, and the kind of impact also will be quite different.
Yeah, just a fragment of the spike protein can able to create the neuronal damage in the tadpole. It is highly risky to think about.
Yes. Yeah, thank you. Thank you for the wonderful lecture.
Thank you, sir. Sir, I'll read out the question from the comment section.
Yes, Dr. Kumar. Happy to receive.
Sir, 1 minute, sir. I just.
Yeah, it's okay, sir.
Happy to visit that. After some, I think 2018 or 19, I met in Malaysia.
I have that photograph, but I was just looking for some old photograph to share the memories. Unfortunately, I can get only from Dr. Jack.
Actually, I got remembered of the past when I see this photo during the olden days in our university, at Madras University. I could remember most of the faces, and I could remember my seniors, everyone, when seeing that photos. Really, it's actually I had a very good time during my PG in Madras University, and Murali sir is my senior actually in Madras University at the biochemistry department. So I have a question. We know that already we have a huge impact of microplastics to the marine environment, and all the species are getting affected, and we have that nearly 400 million tons of more than 400 million tons of microplastics in the marine environment. Again, the face mask, they're saying that it is again a plastic that is thrown, and that definitely it will again end up in the oceans only. It will end up in the marine environment. Again, it is going to affect the marine environment. And also, if it is carrying the same infectious agent, the same infectious SARS-CoV agent, and is there any proof that this SARS-CoV has affected the the fishes, or any proof related to that? Whether any fish, you have said about the tadpole, but any other fish that is from the marine source, any fish got affected with the SARS-CoV? Any Any recent publication related to that, or any in a
As of
summer?
as
For the
of my
marine environment.
Yeah, as of my knowledge, uh, uh, there are reports only saying that trace of SARS-CoV-2 is there in the aquatic environment. As of now, there is no report stating that the aquatic animals are getting affected by SARS-CoV-2. But terrestrial we have, right? We have lions and tigers. But aquatic we don't have yet. And when I say that in aquatic animals we don't have any reports yet, you people need to be more happy because you have
Exactly,
more
sir.
number of projects to be done. So,
Exactly. This will
so
be open venue for us to do our,
yeah,
progress our research
yeah,
towards
definitely it
this.
will open. Definitely it will. As of now, I haven't came across any report stating that there is an effect of SARS-CoV-2 in aquatic.
But anyway, it is go through this animals only, from bat and again, definitely, and also it is affecting many animals nowadays. Maybe in future also the birds and it goes on, it goes on. Thank you, sir. Thank you so much.
Thank you.
Dr. Murali, this is
Thank
in
you, sir.
connection
Thank
with
you for
Dr.
wonderful
Selva's
presentation, sir.
question.
Okay.
Hello.
Yeah, I can hear you.
Yeah, yes.
This is in connection with Dr. Kumar's question. Aquatic environment means is it a freshwater or a seawater?
As of now, I haven't seen anything on the seawater. So it's only on the wastewater, sewage water. Only the report states from here, SARS-CoV-2. And seawater, we are not sure because of high salinity, but even that we cannot able to make sure. Right? Even the seawater has many pathogenic infections, viral and bacterial. So as of now,
Thank
the
you, sir.
reports which I have came across is on the seawater and the sewage water.
Okay, sir. Thank you. Thank you so much.
Thank you for the presentation, sir. I have only one question.
Yeah,
Am I audible, sir?
yeah. Again,
You are audible, sir.
Okay. So, regarding the bacteria, the causes of the coral disease outbreak, yes, sir. The causes of the coral disease outbreaks are complex to understand. Since
yes.
still in the infant stage, there are some fungal-based attacks also observed in the coral reef. I want to know
Yes,
about
there
the
are
fungal-based
different diseases
diseases
like
which are
coral
a causative agent
bleaching,
for the coral.
causative agent for coral bleaching. As you rightly pointed out, there is no single
Bleaching
etiological factor
associated the
for
disease.
coral bleaching.
Yes, sir.
Coral bleaching associated disease. I don't think there will be a disease associated with
Yes,
coral
sir.
bleaching, but thinking in the aspect of ecosystem, as we all know, the calcium carbonate skeleton is going to act as an ecological barrier which can able to prevent from sea erosion. Even we have seen during the time of tsunami, those countries who has the natural the coral reef
Area
barriers
system.
has been prevented from the adverse effects of the tsunami.
Yes.
So that's why even in India it was into the endangered species coral which not disturbed. And of course I don't know why it comes under forestry. That's a funny thing I have heard while I was doing my PhD. While I was trying to collect some
Schedule to run.
samples, I was trying to collect some samples. I went all the way from Chennai to Rameshwaram to CMFRI to get permission to work with corals. They bounced back to Chennai saying that it's under the forestry department, not on the fisheries
Yes, sir.
department.
You should.
Yeah,
Yes, sir. You should get clearance with the PCC.
yeah, that's how it happened. So, but I don't think there is a
Also,
disease associated with coral bleaching. But definitely it will be there because when you disturb some ecosystem, as a consequence, some other disturbance will be there.
yes, sir. Still in our country, the coral reef systems are very close to one. It's not open for research,
Yes.
as you said.
That's
We
why I
need more
struggle.
clearance from the Ministry of
Yes.
Environment. Yes, sir. Thank you, sir. Thank you for the presentation.
Sir, actually,
Yes, sir.
when
Very
we
well
were
put up,
doing
sir.
research,
Yes, sir.
sir, when we were
Yeah.
doing research, at that time we easily collected the marine sponges. Now it is not
Yeah.
possible.
Yeah.
Now it is not possible. It has come into the schedule. Now it is not possible to collect just like that. Even if we get the permission also, we can get only the portion of that. That is very difficult, and we can, yeah, that too, the whatever the things that has separated, or if we found in traces only we can get. It is not that we can directly go and collect it. It is not possible nowadays. So
Yes, sir.
it will be difficult to work
The
on this
non-bailable
marine.
offense, sir. Now.
No, yeah, exactly. So it would be very difficult to work on that. it is quite difficult.
Sarna, I think you can go the question and answer session, which post in the chat box.
Yeah. So there is one question from Prabhakaran. Can we reuse the PCR mix to re-amplify the same DNA template after PCR failure?
We cannot use the same PCR mix after the failure, because when we say failure, it's a tricky word, failure, because failures we don't know whether the reaction haven't happened or our gene of interest is not there. So even if our gene of interest is not there, the Thermus aquaticus, the tag polymerase, is just an enzyme, right? It will be tired after 40 cycles. So if you are going to say, for example, if you want to start running from me, I will be starting energetically. But after running for 5 kilometers, then if you start me to again start with the same energy, I won't have the same energy at the time. So same thing applies to tag polymerase. So tag polymerases are very tired. So it won't do the same efficiency. So it's always better to use a fresh mix for fresh trial.
So next question is, at what temperature DNA will degrade and what prevents our DNA from denature during the polymerase chain reaction?
DNA is a highly stable molecule. Only your mRNA is an unstable molecule. That's why you can remember the movie the Jurassic Park. Okay, so they have taken the dinosaurs' DNA and they can able to clone it after the years. That's not a joke actually. But DNA is highly stable. But of course we can't expect the DNA to be such long. But that's why they have justified in the movie that somewhere the fly has been bitten and taken the blood of the dinosaur that has been into the amber kind of thing. So they can extract the DNA from it. So DNA is a stable molecule that can able to stay longer for years. Your mRNA is an unstable molecule. So whenever you want to do a reverse transcriptase PCR, means you need to take an mRNA, then you need to convert as cDNA. The preference is as soon as you isolate an mRNA, convert it to cDNA. If you keep it as such mRNA, then after like 2 or 3 days, there won't be an mRNA, it will be only an empty solution. So convert it to cDNA, so DNA is a stable molecule. But I don't think there is a criteria to say that how long it is. But comparatively, DNA is a stable molecule compared to RNA. So RNA has to be immediately converted to the cDNA, that kind of good for it. Yeah.
The other question is from Rajini. I would like to know the stability of primers. Will it degrade even under proper storage condition?
Okay, stability of the primers. Nowadays, mostly the primers are supplied to you in a lyophilized format, the powder format. So the stability of the primers will be longer if you store it as such before use. But once you reconstitute with your buffer or with sterile water, then automatically the stability will go on reducing. But even I can't say an expiry date for it. But if you reconstitute, it will say. But even the reconstitute mixture, as the primer is considered to be a DNA molecule, it's stable. It will be stable for a few days. At least I can commit you for a year, at least for 6 months. After that, maybe the quality of the primers can go down, but it doesn't have a fixed expiry date. But if you are ordered now, if you are going to use after 6 months, you can just keep it as a lyophilized powder without reconstitution. After 6 months also, you can reconstitute and use.
So the other question is from Ashwini Suba. Is there any laboratory or company recruiting for PCR? If yes, can you please give some names?
If there is any company recruiting for PCR, means I understand that you want to learn PCR. So while we started our degrees, seeing a PCR machine is very rare. Now we cannot able to see a lab which doesn't have a PCR machine, right? I hope there is a PCR machine. Everybody has a PCR machine. And now because of COVID, like even a diagnostic laboratory, even a small diagnostic laboratory also having a PCR. So PCR machine is available everywhere in the world now. So it's only of your interest to find it out where it is. Once they're readily available everywhere.
Now even the PCR price may come down. It may be available.
Only sir,
Dr. Selvakumar, don't think like that. It has been increased now.
Okay.
It can be possible, yeah,
Maybe high demand.
because of the demand, yeah.
Murali sir, do you think that the nature tells human being, don't think you are mighty. I can control you with a simple virus.
Yes, We are
True,
dependent
sir.
on nature. We as humans depend on nature. Nature doesn't depend on us. Even the human race is going to be vanished from the world like dinosaur. The world is going to be there. World doesn't need human. Only human needs the world.
Accepted. Accepted.
That we should understand.
We selfish. We are selfish. We don't understand. We
Of
never
course,
worry about our next generation, right? That's how the world is going on. So if we at least worry about our own kids, then we won't spoil
Yes.
the environment.
Thank you, sir.
Then,
Thank you
sir,
very much. Yeah, it's very, very detailed and we enjoyed it.
Central officer joins. Good afternoon, sir.
sir, good afternoon, sir. Good afternoon, sir.
Good afternoon,
I told
sir.
sir, good afternoon.
Sir,
He joined very long back. He was very
Yeah, I have
written.
seen
Thank you,
him.
sir. Thank you for coming.
I am really fortunate to listen the lecture like that.
Thank you, sir. Ms. Varna,
Yes, sir.
if
Thank you,
there is
sir.
no question, please proceed with the validity.
Okay, sure, sir. So there is no question from the comment section. I'll move forward. May I now invite Dr. Mutil sir, Controller of Examination, EMET, to deliver the validatory address. I welcome you, sir.
Good afternoon. Most respected Professor K.R. sir, respected speaker Dr. Murali Malliga Raman, and my beloved friend Dr. Senthilnathan, head of the department, and organizing secretaries Dr. Mike and Selvakumar, and other faculty members, participants. Good afternoon to all. I really surprised to two things in this Sunday morning. Actually, today Ahmed is celebrating Away Day program that entirely away from academic activities. We are celebrating Pattimandram and other local things. But the Department of Biotechnology organizing a wonderful event. And another surprise is, being Sunday, more than 100 participants are participated and they are interacted with the speaker as well manner. That is most important things I have to consider here. And that organizing the program also is well, because I past 45 minutes I observed the lectures. He made a wonderful presentation. He multi-dimensional aspects of PCR. That is more important. And also not only but that, he clarified all the questions. I think that the leading India's aquaculture scientist, our Professor Altaf sir, also to agree with my point. He also interacted with our presentation. So that is a really good morning for us. So I thankful to the organizer to organizing this type of events. It will be continuing in the future also. And two things, one more thing I want to represent. That major universities in Tamil Nadu, that Madras University and Annamalai University, the both speakers come from the very good background. They are presented the two type of aspects in well manner. That also we have to be appreciate the department and also the event organizers. And we all know that aquaculture is more important. India have more than 7,000 kilometers of coastal line, but we are still lagging back. Based on the molecular aspects, we have to improve that the breeding technology and species high yield and stress-free varieties like that. So during my college days and PhD times, that we are isolating the Vibrio cholerae from the TCBS agar. That is the most predominant technique. We are using that. How do we have the TCBS agar, NPN technique for E. coli and for SS agar for Salmonella sigilla like that. That time we started that the PCR is the most PCR. We have used that word, but now that all are talking about the fees are reduced like that. But even that the training to be most important for biotechnology students, still we are lacking in Tamil Nadu. We have to improve. Considering this event is well organized. I appreciate the organizers as well as thanks for the opportunity. Thank you.
Thank you very much for the appreciation, sir.
Now I would like to invite Dr. Selvakumar sir, organizing secretary, to propose vote of thanks. Over to you, sir.
Thank you, Sarna. It's a great privilege for me to present the vote of thanks. First of all, I have to thank our Chancellor, Ramachandran sir, for providing the constant support for this, conducting such event. And then I have to thank the Pro-Chancellor, Rajesh Ramachandran sir, for providing the immense support in all aspects. I have to thank our beloved Vice Chancellor, Colonel Dr. G. Tiruvasam sir, for giving permission to conduct such event. I have to thank our Pro Vice Chancellor sir, Dr. N. Manoharan sir, who has also helped in conducting such event. And I also thank our Registrar, Dr. Jayaprakash Veil sir, with his continuous support and advice to conduct such event. This today's event that started with the introductions, welcome speech by Dr. our Senthilnathan sir, our head of the department, followed by the introduction of the speaker by Mayakanan sir, Assistant Professor, Department of Marine Biotechnology. And I have to thank our today's speaker, Dr. Murali Malliga Raman sir, who has been working as a regulatory toxicologist in JLR Laboratories Private Limited. He has said a wide aspect of the PCR and its application, particularly he covered with the aquatic application, that is in aquatic animal health and aquatic toxicology. So he started his speech with the Gary Mullis, who developed the PCR. And also, he also pointed one important thing, that nowadays every layman knows about the PCR. That has come through because COVID-19. So everyone goes for this RT-PCR test now, which is a diagnostic tool that is used to detect whether the patient is affected as positive or negative. And also then he started carried out with his remembering his guru, and even Madam is my mentor, Dr. Nilanja Devraj ma'am. And then he proceed with the PCR thermocycling process and the various phases of the PCR and the detection methods with the dyes and fluorescent dyes, and how to identify and detect a specific sequence in the genome. And also the PCR components and the RT-PCR detection of the SARS-CoV, and the interpretation of RT-PCR result for the COVID. And also he moved to the aquatic applications like the coral bleaching pathogens and the pathogenic expression bacterium at high temperatures, which has some highly conserved regions. And another is the aquatic health, the PCR application in aquatic health, that is microalgae as a feed for fish, and the vectors that carries the growth factors or the vectors that carries the vaccine gene, which can protect the fish from affecting that particular disease. And also the antigenic expression study through PCR gene expression. And again, he explained about the applications of the PCR, that is through gene expression, amplification, screening, and the toxicology studies, that is with the expression of the gene when exposed to heavy metal. What is the specific gene expression? So, and the affected tissue also, how if fish is affected, so there is a gene change, how it affects the tissue. So that can also be detected using the PCR. So how aquatic animals impacted. So we can detect with using this PCR. Then it's the PCR which has a vast application. And also the current, how this COVID-19 or the SARS-CoV will affect the aquatic environment, how it will be affecting the aquatic environment, and how that can be directed or that can be studied using this PCR technique. So he has covered the whole, most of the things, and also the open venues of the research for the aquatic environment. I have to thank Dr. Murali sir, my senior. I have to thank you, sir, for a wonderful presentation. Thank you so much, sir. Over to Swarna.
Thank you, sir. The feedback form is posted in the link box. Sorry, in the comment box. The participants are requested to give the feedback. Thank you.
Thank you, sir. Thank you very much for accepting our invitation.
Thank you.
Even
Thank you, sir.
in the busy schedule, you allocate some time for us to
I
have
really
your
thank you
time.
people to organize it on the Sunday
No,
because I
it
was busy till Saturday.
really so detailed, so detailed. The people who join here really enjoyed. I think they are praising me through the chat box and personally as well. So appreciated, sir. Thank you for your service.
Thank you. Thank you.
We are
Thank you, sir.
fortunate to have
Thank you
you,
so
sir.
much, sir.
Thank you.
No,
Thank you.
thank you, sir.
Thank you.
you.
Thank you all. It's nice. It has a good program.
Thank you, sir.
Thank you, sir. Congratulations, my sir.
Thank you, sir. Thank you very much.
Congrats, sir.
Second event.
Thank you, sir. Thank you very
Call
much.
my senior. You call my senior for this program.
It's good, sir. Sir, you also have to give some talk. We are not going to omit you as well. We know your talent.
We'll do, sir. We'll do, sir.
Thank you, sir.
Don't worry, sir. Every one of us has the chances.
Definitely, definitely. Thank you very much, sir. We have to use it.
Exactly.
Okay.
Exactly.
Dear participant, the feedback link is posted in YouTube as well as in here also. You can fill the feedback and send it to us. The certificates will be reach you within a week time. Okay, after the feedback, we have to get ready your certificate with our PRO assistant. So the certificate going to be reach you in a week time, not immediately. So if you have any doubt, please mail me through the mail which I send in the morning. Thank you very much and stay safe. Okay, there is a couple more events going to conduct by Amet University. Kindly join and get the use of it. Okay, thank you very much for joining.
Okay, my sir. I'm leaving.
Okay,
Thank
sir.
you, sir.
Thank you,
Bye-bye.
sir.
Thank you, sir.
Thank you. Thank you, sir. Thank you.
Again, sir, I'm also leaving, sir.
Yes, sir. Yes, sir. You please, sir. Thank you very much. Yeah.